I can see that we are currently not so many people online, but hopefully we'll have some more people joining us. So, my name is Joe. Can everyone hear me okay? Just write in the chat if you can hear me and if you can see me, please. Let's see, how many people should we have? We have. Can you guys hear me? Let's see what's going on here. Yes, perfect. Let's see. So we're currently testing out this one more window. Perfect. Fantastic. Okay, perfect. So we're just going to wait just one more minute before and just let people join in. So why don't you guys write where you guys are currently live from, just to check that the chat is working and everything. And then also what year of the IB you're in, preferably. Finland, IB1, awesome. Germany. Nice. Awesome. Okay, cool. So we have quite a European crowd. All right. So basically what we're going to be going through today is topic 2, also known as molecular biology. And we'll be going through the main components of topic 2, all based on the syllabus. And we're going to try to make it as focused as possible to try and give you some tips and some tricks on how to basically get your head around this topic. It's quite a big topic. And in HL, it has connections to topic 7 and topic 8, talking about cell respiration and nucleic acid structure and that kind of stuff. It would be nice if we could get to that in the time that we have today, but I have a feeling that we might not get all the way until the end, but we'll get through as much as possible. So how about we just get right into it? So topic 2.1 talks about metabolism. And the first subtopic is to understand that carbon is basically the father of all the different biological compounds that we're going to be talking about. And the reason for this is that it can form 4 covalent bonds. So it has the ability to grab onto loads of different atoms, which means that you can form loads of different compounds. And the 4 compounds that we talk about in molecular biology are carbohydrates, lipids, proteins, and then nucleic acids as well. Okay, so the key learning point here is just to understand that carbon is kind of the important element, and that it can form the 4 covalent compounds. Yeah. It then also, the IB also likes to refer to this concept of metabolism. And the textbook definition of metabolism is the web of all enzyme-catalyzed reactions in a cellular organism. And hopefully you guys will understand what this means. But so enzymes, as we're going to talk about later, are things that can speed up a reaction, right? And so cells and organisms like to have loads of these enzymes so that they can speed up all the different reactions they have to do. And in general, metabolism is split into 2 different subtypes. You can either have anabolism or catabolism. So anabolism is where we take simple molecules and turn them into very complex ones. So we're basically building up. And then catabolism is where we're taking very complex molecule and breaking them into very simple ones. And so the way I like to remember the difference between the two is that someone using anabolic steroids is trying to become very big, right? As you can see in the image here on the left. Whereas a cat would be the kind of person that breaks down a sofa or tears things apart, right? So that's kind of an easy way to remember the difference between the two. And anabolic reactions involves moving from monomers to macromolecules via condensation reactions specifically, Whereas catabolism is about turning macromolecules into monomers by hydrolysis reactions. And so condensation and hydrolysis are kind of the opposites of each other, which is why this kind of makes sense. And so, yeah, that's basically the key parts of metabolism. So throughout the subtopic, you have to refer to loads of different types of biological molecules. And here I've kind of just created an outline of all the different ones you have to be able to draw and recognize for an IB exam. And sadly, we don't really have the time to go into how to draw all these different structures, but it's just, I was trying to give you a feel for what the kind of essential components are. So you have to be able to draw two different types of glucose and a ribose sugar as examples of monosaccharides, and we'll talk about what that means in just a second. You have to be able to draw a fatty acid, and you have to be able to draw an amino acid, okay? And the easiest way to get good at these kinds of things is just to draw them as much as possible. So take out a diagram, take a piece of paper, and then just practice, okay? Oh, and I just forgot to mention, if you ever have any questions, always just write it into the chat, and I'll instantly be able to see that question, so we can stop or move on or clarify things if that's the case, okay? So you just have to be able to recognize and draw these molecules. Okay, urea is another important compound, which you don't have to be able to draw. You just have to understand that it used to be the fact, or people used to believe, that all living things had this magical force to them, which meant that everything that they were made of was somehow special or had this driving force behind it. And the first time in history where scientists were actually able to produce an organic compound in a lab was when they synthesized urea for the first time. And so you have to understand that urea served as kind of a disproving of the theory of vitalism. So the theory of vitalism was what I talked about, with the fact that there was this magical force behind organic beings, which simply just wasn't the case, okay? So that's kind of another compound you have to know about, but not necessarily have to be able to draw or replicate, okay? But that's all for topic 2.1. Does that make sense to everyone? Just so I know that the connection hasn't dropped and everyone's still here. Yes? Perfect. Okay, cool. All right, so 2.2 talks about the structure of water. The structure of water is generally quite simple, right? We know that it's H2O. I'm not going to go through that. And then basically the structure of water is important because due to the orientation of these molecules, of these atoms, so the oxygen and the hydrogen, you have an unequal distribution of electrons. And what this means is that if you have too many electrons in one area, this area is going to have what's called a negative charge or negative pull. Whereas when you lose a lot of electrons, which are obviously negatively charged, then that area becomes relatively positive. And so what water has, which is a very unique ability and which is very powerful in terms of it gives it all of its features, is that it has what's called a positive or a negative pull and a positive pull. And what this allows for is something called hydrogen bonds. And that's a really essential understanding. So we have two different bonds, or two different types of bonds that we can talk about in water. We can talk about the intramolecular bonds, which are the bonds that actually bond together the atoms of hydrogen and oxygen. And then you can also have something known as an intermolecular bond, which is a bond that exists between the different water molecules. And I hope this is hopefully making sense to everyone. And what's key about these hydrogen bonds is that these are quite hard to separate. When we talk about under the category of intermolecular bonds, hydrogen bonds are quite strong. So it takes a lot of energy to separate these bonds. And as we're going to talk about, water has a lot of properties which arises from just this principle, okay? And later on, when we talk about these properties, anytime you get an IB question which asks you to explain one of the properties, always refer back to the structure of water, talking about how water has a dipole, which allows for hydrogen bondings and strong interactions, okay? Okay, so there's two characteristics or two ways to describe the ways that things can interact with water. So things can either be hydrophilic or they can be hydrophobic. When something is hydrophilic, what that means is that it is attracted to water. So it likes water. That's kind of what the philic means at the end, okay? And something hydrophobic is something that does not like water. Now, in very simple terms, hydrophilic things, which are things that you can see on the left, are things that have some kind of charge. So they'll be attracted to part of this water molecule. They'll either be negatively or positively charged or have some kind of a pull. And so that means that they're going to be attracted to water. Other things which don't have a charge, for example, fats, they'll have nothing that they're attracted to. And so that's why when you try to mix oil with water, because fats are hydrophobic, they're not going to have an attraction. Okay? And so yeah, these are just two words that you're going to be using all the time in biology. And so it's really key to get the understanding. Let me just see this question here. You do not need to know the name of the scientist who disproved the theory of vitalism. There is a Miller-Urey experiment, which we don't go into here. It's just to answer your question, I promise. But we sadly don't have time to go into that now. But you just have to understand that there was this Miller-Urey experiment. Okay, you can maybe, yeah, okay, fantastic. Okay, right, so we can have hydrophilic and hydrophobic things. Okay, but so what are these properties of water that we've been talking about? You have to be able to list 4 of them. And it's a very popular topic that comes up in IB exams. And yeah, so the 4 of them are cohesion, adhesion, thermal properties, and solvent properties. So what is cohesion? Cohesion is when 2 things that are the same stick together. So when 2 water molecules have hydrogen bonding between them, then this attraction will make them stick together. So water is basically relatively sticky to itself, which is what you see in this image here, where these droplets, they kind of all accumulate together. They don't want to spread out. Adhesion is the fact that water is attracted to anything that's polar, which makes sense. So anything that's hydrophilic is going to have an adhesive property to it with water. The third thing is thermal properties. And again, always make sure to, when you're explaining these things, refer back to the structure of water. So because water has this dipole and it can form these hydrogen bonds, which are very strong attractions to itself, if you were to cover someone with water, then the body's heat will naturally be used to separate these water molecules from each other. And because it takes a lot of energy to separate these water molecules, water acts as an excellent coolant. Right? So the reason why we cover each other with, or why we cover ourselves with sweat when we're very hot is because our body's trying to cool ourselves down, right? And so if we're covered in something which takes a lot of energy to evaporate, well, then that's a really good and efficient way to cool the body down. So it's a natural mechanism in what we call homeostasis, okay? And then the last thing is solvent properties. And again, this still refers back to the dipole across the water molecule. It's just that water, if you immerse something into water and it is polar and it's able to be separated, well, then water is going to dissolve that those particles. And so water is known as a universal solvent because it can be used to basically dissolve most things that are polar. Yeah? Make sense to everyone? I will take the silence as a yes. Okay, so you also have to understand that because of these different properties of water, water becomes quite useful in biological organisms. So it has cohesive properties, which allows for something like transpiration and the Jesus-like capability of insects, right? So the fact that insects can walk across water is because water molecules like to stick to themselves, right? So they don't get broken very easily. The adhesive properties also help in transpiration, which comes up in topic 9 for HL students. In the thermal properties allows for sweating, as we've already talked about. And then the solvent properties allows for blood plasma to dissolve nutrients, right? So we can carry nutrients around the body in our blood. And then again, also for in plant tissues to transport nutrients in the phloem and the xylem. Mainly in the phloem in terms of nutrients, but we can dissolve things in the xylem as well. Okay, so just knowing that because of water's structure, it has specific properties which makes it really useful in biological organisms. Okay, and that is basically the end of water. So the next thing we have to talk about is carbohydrates. Are there any questions at this point, or anything that doesn't make sense? No, fantastic. Okay, cool. So carbohydrates. Carbohydrates are split into 3 different classes. We can call them monosaccharides, disaccharides, and polysaccharides. And since I'm from Denmark, I like to think of things in terms of Lego blocks. So monosaccharide is just if you were to have an individual an individual Lego block all by itself, one individual unit of carbohydrate. A disaccharide, the di might give away, is when you can combine 2 different Lego blocks together to form a unit of 2 individual Lego blocks. So 2 monosaccharides put together form a disaccharide. And then a polysaccharide is when you combine loads of different monosaccharides together to make a much more complex structure, such as this beautiful Lego house here. Okay? And you have to know examples of each of these categories of carbohydrates. So monosaccharides includes things like galactose, glucose, ribose, fructose, right? And you just have to be able to remember these. There's not too much to it. Disaccharides are things like maltose, lactose, and sucrose. And then polysaccharides, it's about the 3 main examples you have to know are starch, cellulose, and glycogen. And we're going to go into a little bit more detail about those in a second. Okay, so carbohydrates have 3 different classes. You have to be able to describe how different polysaccharides and disaccharides are made up of monosaccharides. Okay, so the 3 disaccharides that you have to know are sucrose, maltose, and lactose. And sucrose is made up of glucose plus fructose, 2 alpha glucoses and a fructose. Maltose is made up of 2 alpha glucoses put together, and then lactose is made up of beta glucose and a galactose. Now, the way that I like to remember these 3 different ways is that maltose, you just have to kind of memorize that it's 2 glucoses put together, but that sticks quite well because it has a lot of references in the entire syllabus. Sucrose, the way to remember what its structure is to remember that fruits are sweet. So as you can see, all the disaccharides are composed of at least 1 glucose. So the actually important thing to remember is just what else is combined with the glucose. And so since fructose sounds like fruits and sucrose is kind of like sweet or sugar, and those 2 things kind of work together. So maybe that will help you. And then galactose, lactose, the way that I like to remember what the structure of lactose is just to think of the Milky Way chocolate bar. I don't know if you know what that is. But so the Milky Way chocolate bar is kind of this chocolate that has like a white core. And Milky Way kind of refers to the galaxy, right? And the core of this Milky Way chocolate is lactose. Okay, so maybe because those things are stupid enough, you might remember them. I don't know if that helped. The polysaccharides, starch, glycogen, and cellulose. Starch and glycogen have very similar structures, and they're both composed of alpha glucose. Okay, so we're going to talk about the slight difference between them, but those should be pretty straightforward. And then cellulose, so the thing that makes up the plant walls, is made up of beta glucose, and we're just going to get into those structures. Okay, but so the key thing here is just, sadly, you just have to memorize what the different components of the disaccharides are. Okay, so we're going to go into a little bit more detail about these polysaccharides. So one thing that you have to understand about each of them is that they serve for a different function. So in plants, starch serves as a method of energy storage. And in animals, their way of storing energy is glycogen. So as I said earlier, these 2 have a very similar structure, which is also why they have a very similar function. Okay, so they both act as energy storage. And cellulose is used by plants to give them structure because what their cell walls are made of, right? So in topic 1, you know how you talk about eukaryotic cell structure and how plant cell walls or plants have cell walls. These cell walls are mainly composed of cellulose. And so they provide structural support. Okay, so you just have to be able to list these 3 things out. Okay, so what is the difference or what is the structure of starch and glycogen? We've said that they're quite similar. So starch is, as we said, a polymer of alpha glucose. And these glucoses can be linked in 2 different ways. Either they can be linked in simply just a straight chain, right, by what we call 1,4 glycosidic bonds. And this is what forms kind of the subcategory of starch that we call amylose. Okay, so they're just formed by 1,4 glycosidic bonds. That's just kind of a conceptual understanding. What can also happen is that these glucoses aren't just linked in a straight chain, but they actually also form a branch. Okay? But in order to form this branch, you have to introduce a different type of bond between the two monosaccharides. You have to have 1,4 bonds as you always do, but then you also have to have what's called as a 1,6 glycosidic bond. And when you introduce a branch into what was amylose, the molecule becomes amylopectin. Okay, so starch and glycogen are both composed of amylose and amylopectin. The difference between glycogen and starch is simply that the amylopectin in glycogen has a lot more branching, because it has to be able to be built up and broken down much more quickly. But other than that, it's basically a very similar structure. So just remember, glycogen has much more bonding, and hence it would have much more 1,6 glycosidic bonds. Okay, make sense to everyone? I can see that we are not too many people online yet, but that's okay. I'll assume that you guys, the frequency of 1,6 bonds, exactly, yeah. Fantastic. Okay, so what about the cellulose structure? Well, so cellulose is made up of straight chains of beta glucose, right? So it's formed by 1,4 glycosidic bonds as well. The reason why cellulose provides cellular structure is that these kind of sheets of cellulose can be layered on top of each other, and you can then create hydrogen bonds, the same that you had in water, between these two different layers, which means that you basically have it's kind of like sheets of paper that have glue on them on both sides. If you put loads of those together, that makes like a solid textbook with glue in between each page. It's quite strong, right? And so that's why cellulose is quite powerful, because these hydrogen bonds, when spread over a large area, provides a lot of strength for the plant. Okay, so you have to be able to describe the starch and glycogen structure as well as the cellulose structure. Okay, and that's it for carbohydrates. So any questions here before I move on to lipids? Nope. Okay, fantastic. Okay, so fats and lipids, or sorry, all biological molecules are composed up of monomers. We said that in carbohydrates, that monomer, so that individual unit, was a monosaccharide. In lipids, the individual unit for lipids is what we call a fatty acid. And the basic structure of a fatty acid is the one that you can see up here at the top. So where you have, so you'll have what's called a carboxyl group at the end on the right-hand side over here. And then you just have a straight chain of carbons, and then a triple bond on the carbon, or not a triple bond, but 3 bonds coming off of the carbon at the end, okay? And that's what we call a saturated fatty acid. Does anyone know why it's termed a saturated fatty acid? Any idea? Okay, well, so a saturated fatty acid, well, the reason why it's called a saturated fatty acid is if you think of something saturated, it's something that can't have anything more put onto it or put into it. And in terms of fatty acid, what we're talking about is the hydrogens, okay? So a saturated fatty acid is something where you cannot introduce any more hydrogens onto the carbon chain. And then, of course, you can have an unsaturated fatty acid. And the way to create an unsaturated fatty acid is just to create a double bond in between the carbons. Because the only way that you can do this, right, if we remember carbon can only form 4 bonds, is to remove one of the hydrogens from the carbon chain. And so by doing this, you're not at your full potential of hydrogen, so you're what's called unsaturated fatty acid. These unsaturated fatty acids have 2 different subcategories. Things can either be cis or they can be trans. So a cis fatty acid, what this basically means is that the 2 hydrogens that are present across the carbon double bond are on the same side. So as you can see here, the hydrogen on this carbon is on top of it, whereas the hydrogen on this carbon is also on top of it. And the way to remember what a cis fatty acid looks like is kind of remember that this structure would form a C, or a C that's facing upwards, okay? The other type of unsaturated fatty acid is a trans fatty acid, which is where instead of having a C, you have something that looks a little bit more like a T. So that might be useful to remember in that way. Okay, but so these are just subcategories of the fatty acid. Okay, so you just have to be able to draw each of them. Okay, so how do we form the macromolecules in the lipid structure? One of these macromolecules is what we call a triglyceride. And triglycerides are formed when we form condensation reactions between one glycerol molecule and 3 fatty acids. Okay, so what you see in this diagram here is on the right-hand side is a glycerol molecule, which is just a straight chain of carbons with hydrogens on one side and then hydroxide groups on the other sides. Yeah, so that's just the basic molecule of this glycerol. And then to form a triglyceride, you basically just have to attach fatty acids. So this OH end of each of the molecules condenses to form water. And what you're left behind with is something that looks like this, where you have a carbon double bond and then a carbon oxygen single bond and then another carbon. Okay, so this is what we call an ester bond, right? And you have to be able to remember that. To form specifically a triglyceride, you have to attach 3 fatty acids to the glycerol chain. If you were to just attach one fatty acid chain, does anyone know what that would be called? Exactly, so that's what we would call a monoglyceride, yeah? So one individual fatty acid creates a monoglyceride, but 3 of them creates a triglyceride, okay? But so these are formed by condensation reactions and they produce what's called an ester bond. Okay, so another subtopic in terms of lipids is to understand the ability to store energy relative to carbohydrates, okay? And it's specifically to carbohydrates. And as you might know, the main way that humans or animals, specifically humans, like to store energy is via depositing fats, right? And the reason for this is because fat is a much better energy storer than a carbohydrate is. Because if, for example, you look at 1 gram of fat, that contains 9 kilocalories, whereas 1 gram of carbs contains only 4 kilocalories. And so if you imagine like the caveman back in his time who had to carry all of his energy on him at all times, right? Because he wasn't just able to eat and go to McDonald's whenever he wanted. The most efficient way to carry energy, since you have to carry it around, is having the most amount of energy in the least amount of weight possible. And so that's why evolutionarily, people really like to contain energy in the form of lipids. Okay, so it's the most efficient energy storage because it has the highest density of energy. Yeah, so the most amount of energy per mass. Okay, so how do we assess people's relative health, I guess you could say, with regards to lipids, is something called the body mass index. And the body mass index is just a way to standardize how much people weigh compared to how tall they are, right? Because if you have someone who is this tall and someone who's this tall, and both of them weigh 100 kilograms, that doesn't mean that doesn't equally compare in terms of how healthy they are, right? And so what you want to do is that you want to refer them onto the same scale. And the way you do this is using BMI. So one type of BMI calculation is using something called a nomogram. And you have to be able to use this on an exam. And basically, the way it works is that there's 3 lines or 3 scales. On the very left-hand side, you'll have a scale for height. I think that's what it says here. No, sorry, a scale for weight. And there you basically, the way it works is that you just plot whatever weight you're looking at when considering the BMI of a person. So let's say a person weighs, what does this say here? Weighs 80 kilograms, right? And then on the other side, you'll have the person's height. And so you plot whatever height there is. And then using a straight edge, you basically just connect these 2 points. And that's going to intersect at some point on this BMI scale. And so you just read off whatever the BMI value, and that's your BMI. Okay, and you actually have to be able to use this nomogram for IB exams. So it's important to kind of remember that. The other way, which is kind of the more standardized way, and the way that the computer age has kind of brought along, is to calculate BMI using a formula, which is your weight in kilograms, or your mass in kilograms, divided by your height in meters squared. So for example, if you were 2 meters tall and weighed 100 kilograms, it would be 100 divided by 2 to the power of 2, aka 4. Okay, so you'd have a BMI of 25. Okay, so you just have to remember these formulas and how to use the nomogram. But that is pretty much it for lipids. I would like to move on to proteins if there are no questions in the audience. Okay, fantastic. Okay, so proteins. Here's where things start to get quite important. So if there's any subtopic in topic 2, which I think is essential for any other topics, it would be the protein subunit. Because it's really important to understand the structure of amino acids and how they link together, because it refers to how our genes are built up and how our body functions, okay? So the monomer, when we talk about proteins, is the amino acid, as we've already mentioned, right? And you can link together multiple amino acids with condensation reactions, because as we remember, all anabolic reactions are condensation reactions, right? And so this is the basic structure of an amino acid, where you have an NCC, so a nitrogen and 2 carbons. You have 2 hydrogens on the nitrogen, which forms what's called an amine group. And on the carbon, you have this double bond to an oxygen, and then what's a single bond to a hydroxide group. And this forms what's called a carboxyl group. Now, the thing that I just want to emphasize, which might be quite basic for some of the HL people around, is that the carbon has, or the R group that is bonded to a carbon isn't actually an element in itself, but rather it represents a variable group. So this is something that can change in an amino acid. Okay, so you can have many different types of amino acids, and that what varies is the R group, nothing else. Okay, and so when you link 2 amino acids together, you form what's called a dipeptide, and when you link many more amino acids together, you get what's called a polypeptide. So it kind of follows the same logic as monosaccharides, disaccharides, polysaccharides, right? It's just that now we're talking about peptides. Okay. Right. So in total, there are 20 amino acids, which means that you have 20 different R groups possible for any given amino acid. Okay. And you can link these amino acids in any different sequence in order to create a polypeptide, right? And so if you consider how many amino acids we have in a specific polypeptide, you can then work out how many actual combinations there are. So if you had a polypeptide with 10 amino acids, what that means is that for every position, you have 20 possible amino acids, meaning that the total number of combinations you can make with just 10 amino acids is 2 to the power of 20, which is a ridiculously high number. And the reason why I included that is because you have to understand that just this tiny, even though there's only 20 amino acids, this allows for a massive amount of variety in terms of the amount of proteins or the structures that you can build, okay? The thing in the cell that actually forms this linkage between amino acids is something called the ribosome, which you'll definitely have heard about. And yeah, basically what ribosomes do, as we're going to talk about, is just link these or form these peptide bonds between amino acids in turn to produce proteins in the end. Okay? And yeah, that's basically the learning outcome for that. Okay, but so how does a ribosome know which sequence of amino acids it's supposed to link? Well, basically, you can think of the DNA or the genes as the dictator of the amino acid of the ribosome, right? So it provides the instructions to the ribosomes using the genetic code. Very straightforward. Okay. Proteins also have. We can describe the structure of proteins using specific language in the IB. And in general, proteins can be described under 4 different categories. You can have what's called the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. And you have to be able to describe what the difference is between all these structures. So the primary structure of a protein is basically just describing which amino acids have a link together to create this specific protein. So if I have a protein that's made of, let's say, 100 amino acids, then, well, which amino acids are they? Is it glutamine, valine, whatever, right? So that hopefully makes sense. So that's just a single chain. When we talk about secondary structure, we talk about the initial folding that this primary structure does because of the interactions that the different amino acids have with each other. And what does that mean? That means that different amino acids will have different attractions to each other, right? They can do polar attraction, they can do hydrogen bonds, all these kinds of things. And because of this, the protein will start to fold out of its primary structure, and it can fold into two possible structures. Either it forms what's called an alpha helix. So let me just write that out so you can see it. It either forms an alpha helix or what's called a beta pleated sheet. So these are the two possible forms of secondary structures in proteins, right? Yeah, that's basically it. So it's just these attractions between amino acids. These secondary structures can then get even more complicated, because after the protein is folded and formed this alpha helix or beta pleated sheet, what it realizes is, well, there's still some attractions between different parts of this alpha helix or beta pleated sheet. So what happens? These attractions will want to come together. So again, polar interactions or hydrogen bonds will mean that the protein again creates folding. And a tertiary structure refers to the fact that this is the first time that a protein folds into 3D conformation. So you would be able to draw an alpha helix or a beta pleated sheet on a piece of paper, but you wouldn't accurately be able to draw a tertiary structure because it's a 3D thing, right? So that it gets a little bit more complicated there. But again, this is just because of the interaction between amino acids. And then a quaternary structure occurs when you have multiple 3D structures that then come together. And when we have a protein that's made up of many individual pieces of, or many individual chains of polypeptides, we call these individual chains a subunit, okay? So for example, hemoglobin, which is the thing that carries oxygen in your red blood cells, is made up of 4 subunits, which means that it's made of 4 individual polypeptide chains. It's made up of 2 alpha and 2 beta polypeptide chains, but that's not super important right now. The important thing to understand is that these are separate chains that have folded and they're then attracted to each other to then form a more complex molecule. Let's see, we have a question. So alpha helices and beta pleated sheets are secondary structures, okay? Yeah, so it's just this secondary type of conformation. We have primary, secondary, tertiary, and quaternary structures. And an example of quaternary structure is hemoglobin. Fantastic. Okay. So you also have to understand what or how we can collect all the possible proteins we can create in a human body. And that wording might be a little confusing, so let's kind of break it down. So we said earlier that the genes in our body determine which proteins we're able to create, right? And the genome is the sum of all the different genes that an individual cell or an individual organism might have, right? And so if you were to sum together all these different genes and then look at which proteins can all these genes create, well, then all the proteins that you can create is what's called a proteome, okay? So I guess that kind of links together, right? A genome codes for the proteome. And so the proteome is a unique set of proteins that is found inside every individual cell or body, and more specifically the body, for an organism, okay? And you just have to understand the definition. There's not much more to it. You also have to understand that different proteins have different functions, and the IB specifically wants you to know these 5 examples. So ribisco, insulin, immunoglobulin, rhodopsin, collagen, and spider silk. There's not much more for me to say about this other than the fact that you just have to memorize what each of their functions are. I would recommend trying to make some cool story out of it or reading up a little bit about each of these proteins, as the more knowledge you have about something, the easier it is to memorize. So kind of just as a tip, just to read into it a little bit more, learn something about immunoglobulins or rhodopsin or collagen, and that will probably help your revision. Okay. So the difference between a tertiary and a quaternary structure is that a tertiary structure is just made up of one polypeptide. It's just one peptide that has folded into 3D structure. But a quaternary structure is when you have multiple polypeptides, so multiple chains, that then come together because of interactions. Okay, so they look similar, but the quaternary is made up of multiple peptides, whereas one. Okay. Fantastic. Yeah, a tertiary structure can still be a functioning protein. Yeah, you don't have to be a quaternary structure. That's exactly right. Okay, so we said that we said that proteins are composed of the interactions that they're made up of, right? The sequence of amino acids will determine which type of attractions you can find, which then determines how the proteins folds, right? What you can do is that you can break down proteins. You can ruin the way that they're formed or the way that they're currently set up. The way to do this is simply just by breaking the bonds or by disturbing the bonds that has created this folding in the first place. There's two ways to do this. Either you can introduce or expose the protein to very unusual pH, because you don't have to know what exactly pH means other than the fact that it will mess with these hydrogen bonds that exist inside a protein. Okay, so if you give weird pHs, you'll have weird attraction to the protein, so the protein falls apart. Similarly, if you expose proteins to high temperatures, the molecules in the protein will start to move more and more and more, right? That's the definition of temperature. And so as that happens, the bonds that exist between the molecules, not the molecules, the amino acids, they're going to have a harder time holding this structure together. And so eventually protein falls apart. So what does this mean in terms of real life? Well, when you take an egg, right, and you look at it raw, the structure is quite see-through, right? Like you can quite easily see through an egg white, right? But if you expose this egg to a high temperature, for example, by frying it, all of a sudden, the protein inside the egg, which is a protein called albumin, that protein starts to break down. And all of a sudden, it's not going to be see-through anymore. It's going to be tough and white. So that's why when you fry an egg, the thing goes from being quite clear to then being quite see-through. But what you might not have done is actually put an egg inside a weird pH. So if, for example, what you can do is you can take an egg and you can put it inside a glass of vinegar. And what happens there is basically the same thing as what happens when you fry an egg. The thing will become white. And this is because you are breaking down the protein structure. Okay, so this process is called denaturation. You're denaturing proteins by exposing it to weird pHs or weird temperatures, specifically high. All good with everyone? Fantastic. Okay. Okay, so the next thing that we have to talk about is a specific type of protein known as an enzyme. So what are enzymes? Enzymes are what's called a biological catalyst. And a biological catalyst, or a catalyst in general, is anything that can speed up a reaction without itself being broken down by the reaction. So it has to be in the same state before and after the reaction has occurred. Okay? So how does an enzyme act as a biological catalyst? Well, the way it does it, it has a specific area on it that we call the active site. And when this area specifically collides with the substrate, which is the thing that's reacting, then the active site can kind of change the chemistry of whatever reaction was going to occur. And in the process, the reaction is sped up. Now, the way that we change the chemistry is by lowering what's called the activation energy. So as you can see in this diagram here, an activation energy is the energy that any particle has to have in order for a reaction to occur, right? Which might sound a little bit abstract at first, but if you just imagine energy levels being hills, then the initial activation energy, the initial energy that a particle had to have in order for a reaction to occur, can be represented by this gigantic mound here, okay? And what an enzyme does is that it simply brings this down to a lower state, meaning that more particles are going to be able to have this enough energy to react, and hence the reaction is going to happen faster. Can anyone tell me why it might be useful to have enzymes in the biological organism? Why don't we just let reactions occur all by themselves? I guess we don't have enough people to really make that a big discussion. But yeah, so the main thing is that enzymes speed up a reaction. Okay, they don't allow for reactions that would never have occurred in the first place to occur, but they do make things go faster. And so because the cell is constantly doing different reactions, it's much more beneficial to have them occur really fast, right? And so what you have to know is that enzymes are biological catalysts. They collide with their substrates on their active sites, in the process lowering activation energy, and so speeding up how fast a reaction can occur. Okay? So there are different factors that affect how fast an enzyme will work. And you just have to understand how each of these affect enzymes. So the first thing is temperature, which we're going to represent by this top graph here. So as I increase, imagine the x-axis being temperature. Sorry, let's ignore that for here. Imagine temperature is down here, and rate of enzyme is up here, right, on the y-axis. As I increase temperature, the molecules that the enzyme is working with are going to be moving faster, right? And we said earlier that in order for a successful enzymatic reaction to occur, the molecules have to collide, or the substrate have to collide with the active site of the enzyme. So that means that if the molecules are moving around a lot faster, there's going to be an increased chance that this collision occurs. So as temperature increases, you have an increase in the rate of enzyme activity. Okay? But then what also happens is that at a certain point, all of a sudden the rate of enzyme activity starts going down again. Now, why is that? Well, the key thing to remember is that enzymes are just proteins, right? And so if you increase the temperature of a protein, as we've already talked about, you're going to denature the protein and the enzyme. So at a certain point, you start changing the shape of the active site. So whereas it used to look like this, after a bit of heating, it might look like that. And so whereas a substrate used to be able to fit into this reaction, now all of a sudden, there's no more attraction. And so all of a sudden that slows down the reaction. And so the rate of enzyme activity goes down. Okay, so there's basically two parts of this temperature graph. What about pH? Well, pH is in a very similar way to temperature, except that we have an increase in activity as we approach what's called an optimal pH. But then we also have a downfall. And the reason for this isn't because of the reason why we have an increase in the rate as we approach whatever given pH is that proteins are denatured as we move away from the ideal pH, right? We talked about that earlier. The fact that if you change the pH, if you introduce extremes into the pH, the protein will break down. And so as you change pH, the rate of enzyme activity simply decreases, like this. And so yeah, that's exactly right. So basically, if you move away from the ideal pH, then the protein denatures and so the rate of enzyme activity goes down. And then the final thing is substrate concentration. And you might be able to work this one out, or you might already know it. But basically, if my reaction is dependent on how fast my molecules can collide with my active site, well, if I have more molecules, right, then obviously there's going to be much higher chance of a successful collision to occur. And so as I increase substrate concentration, my rate increases. But at a certain point, what happens is that this graph levels off and there's no more change. No matter how much I increase substrate concentration, the rate doesn't increase anymore. And why is this? Well, that's basically when your enzymes are working at full capacity. They cannot take in substrate at a faster rate. So it doesn't matter how much you increase substrate concentration. The enzymes are already doing their best. So you can't increase rate beyond this point. Okay? And so what you have to be able to do is just describe each of these graphs, draw out each of these graphs, and then explain why the graphs behave this way. So did everyone just lose my screen? Okay, let's see. We have a question over here. Okay. So I was drawing something. Did that not get projected at all? That is a shame. Okay. Yeah, I was drawing grass this time. Oh, I'm sorry. In order to draw, I had to go full screen, so that's probably what the problem is. Basically, the grass looked like this. It's, yeah. Okay, sorry about that. Right, but so that's basically the end of enzymes, okay? So you have to understand how these different factors affect the rate of enzyme activity, and you have to understand what enzymes are and the fact that they're proteins, okay? The next thing would be to talk about the structure of DNA and RNA. But does anyone have any questions at this point? No, there are no questions. Fantastic. Okay, so DNA and RNA. These are the things that kind of make up our basic structure, right? The structure of a genetic material. So what is the basic unit of DNA and RNA? Because as we've talked about, all complex molecules basically are made up of monomers, right? They're made up of individual units. And in terms of DNA and RNA, the monomer, the basic structure, is what we call a nucleotide. And a nucleotide is composed of three things. It's composed of a phosphate group, which you can draw as a circle. It's made up of a pentose sugar, right, which just means that it has 5 carbons on it. And then it has a base, okay? And DNA and RNA both have this basic structure. The only difference is that DNA, the bases are guanine, adenine, thymine, and cytosine. Okay, those are the possible bases. RNA has the same bases, guanine, adenine, and cytosine. However, their thymine is replaced by a uracil molecule. Okay, so it's just the same basic structure, it's just that there's a slight difference. Also, in RNA, the pentose sugar has all of the oxygen on it that it would have if it was a normal ribose molecule. Okay, but in DNA, there's an oxygen missing. And that's kind of given away by the name of what the name of DNA actually is, because DNA stands for deoxyribonucleic acids. Okay, it's an RNA molecule, some called nucleoside triphosphate. Typically, we just call it the base. Okay, in terms of the IB. Okay, so these different, we just call it a nucleotide. Typically, you don't have to worry about the triphosphate part. Yeah. Okay, so these different monomers can obviously form more complex structures, basically strands of nucleotides. These can form what's called a phosphodiester bond. And this bond exists between the phosphate group and the third carbon on the ribose sugar. Okay, and so as we create this link of nucleotides using phosphodiester bonds, what you end up with is that one end of the molecule is going to start off with a phosphate group, and one end of the molecule is going to end with basically an exposed third carbon. And we give this specific names, which are really important for the rest of the topic. And the top of the chain with the phosphate group is what we call the 5' end, whereas the end with the ribose is called the 3' end. Okay, and that's pretty important to understand what the difference is. And nucleotides can only be added from a 5' to a 3' direction. So that means that if I were to add another nucleotide to this chain on the left-hand side, I could not add one up here, but I could add one down here, right? And so by forming these links, then yeah, that's how you build DNA, okay? DNA specifically is not just made up of one strand of nucleotides linked together, but it's also made up of another strand, which kind of is bonded to this strand, but not by an intramolecular bond, but rather by an intermolecular bond and by our favorite hydrogen bond. Okay, so as you can see, hydrogen bonds are really important. They come up all the time. So, and hydrogen bonds exist between specific nucleotides. Because hydrogen bonds can only exist, well, generally speaking, the bonds that exist between nucleotides always follow one specific pattern, what we call complementary base pairing. And this describes that if there's a nucleotide with a G base, that can only form a hydrogen bond with a nucleotide with a C base. So G always goes with C and T always goes with A. I'm sure that has been taught to you many times in the course of your biology career, but now you just get a recap of it. Okay, so that's what we call complementary base pairing. That word is quite important to remember. Okay, and the strands that link together are always run in anti-parallel. And what that means is that if there's one strand which runs from a 5' to 3' direction, the other strand has to run 5' to 3' in the opposite direction. Okay, so those are kind of just two key learning and understanding points. But there's not much more to it than that. Okay. Right, so we can do quite interesting things with these different nucleotides. Now, in HL biology, you go into a lot more detail than you do in SL biology, right? Now, because this, for simplification's sake, and since we're only going through molecular biology, I'm basically just going to be taking you through the basics of how replication, transcription, and translation work. Okay, so if you're HL, you're going to have to know this in a lot more detail, and I'm sorry for you. But in terms of SL, things stay quite simple. Okay. Okay, so DNA in SL, you have to understand that DNA replication is semi-conservative. Now, the people who discovered this are these two guys down here, called Mieselson and Stahl. And what they basically realize, and what they basically describe by this term semi-conservative, is that when DNA replicates, each parent strand of DNA is going to produce two strands where half of the genetic material is old and half of the genetic material is new. Okay, so it's not like the DNA is completely copied and all of the new DNA is made up of new genetic material. No, DNA is split and then forms kind of the template for the new genetic strand. Okay, and so over time, basically the parent strand will get more and more diluted. But for every parent, you get half, in each offspring, you get half of the genetic material in each. Okay, that's what we call semi-conservative. So DNA replication in the very, very, very simple steps. So DNA replication starts off with this beautiful enzyme called helicase. And what helicase does is that it basically takes this double helix of complementary base paired genetic strands and it unwinds that helix. Okay, it unwinds it and then it separates the hydrogen bonds that exist between the two different strands. Now, why does it do this? It does this to expose the nucleotides that sit or that are kind of on the inside of this double helix, right? And as you expose the insides of the genes or of the DNA, what happens is that this other enzyme called DNA polymerase can suddenly come in and it can attach new nucleotides to that parent strand, okay? And it does this in a 5' to 3' direction, okay? But so the old strand is basically a template because all the DNA polymerase does is just it takes a nucleotide that's floating around in the cytoplasm and it sees, oh, okay, here I have an A nucleotide on my template strand. A only fits with T, so it selects a T nucleotides from the cytoplasm and then allows for them to do complementary base pairing and then forms a phosphodiester bond between the nucleotide that is right next to it, okay? So DNA replication can be simplified to two very simple steps. Helicase unwinds the double helix and separates the 8 hydrogen bonds, and then DNA polymerase can link new nucleotides together, forming a new strand from the old strand, okay? But the old strand will act as a template. Is that clear to everyone? Fantastic. Okay. So we said earlier that the thing that codes for proteins is something called the ribosome, right? And we also said that the thing that has the code for whatever protein we want to produce is the genetic material DNA, right? The problem is that ribosomes are found in the cytoplasm of the cell, whereas DNA is found in the in the nucleus of the cell, right? So how do we create this communication between a ribosome and the DNA? The way that we do this is with something called mRNA, or messenger RNA, right? So it does what it says on the tin. The messenger is basically because it carries information from DNA to the ribosome. Okay, and this process of forming mRNA is something called transcription. So transcription is the making of mRNA from DNA. Now, it is done by an enzyme called RNA polymerase. And what it does is that it basically just moves along whatever DNA strand it wants and then separates these two strands apart. And as it separates the two strands, it can basically add new mRNA nucleotides, or RNA nucleotides, to form what's called an mRNA molecule, right? And it also does this from a 5' to a 3' direction. But after RNA polymerase is done with a specific gene, then what you're left with is just a simple strip of mRNA molecule. And this mRNA can then do a bunch of stuff and then eventually end up in the ribosome, where it can share its information. Okay, make sense, everyone? Awesome. So yeah, this here is just to replicate the fact that because of complementary base pairing, each DNA strand is going to have a specific RNA strand that it wants to match. And this is universal, right? Okay, so what do we do after we've actually taken a copy of this genetic code and we've brought it to the ribosome? Well, then we actually have to have a way to produce proteins from this mRNA molecule. This process is called translation. And in very simple terms, it's all it is just making polypeptides on ribosomes. The way the DNA is built up is that we're only going to do SL today. Yeah, sorry, because the HL part isn't molecular biology. It's topic 8 and topic 9. Okay. So the way that the DNA is built up is that 3 bases, so for example, CUC, corresponds to what's called 1 codon. And 1 codon codes for specifically 1 amino acid. And so you can imagine if you have 27 bases in a row on an mRNA molecule, that corresponds to 9 different codons, which corresponds to 9 amino acids linked together. Okay, so 3 bases equals 1 codon equals 1 amino acid. And each specific base sequence codes for an amino acid, as we're going to talk about. Okay, but so how does the ribosome link together amino acids using this code? Well, it uses something known as tRNA. And tRNA is also a strand of RNA, which has this clover-leaf structure, as IB likes to call it. But what it also has is an amino acid attached to the 3' end. And what it can do is that there's all these tRNAs floating around in the cytoplasm, which contain specific amino acids which correspond to specific codons, right? So each tRNA has a specific region which determines which amino acid it has, okay? Now, because each amino acid is specific to a code, that means that when the tRNA comes close to an mRNA molecule on a ribosome, right, because the ribosome is the thing that reads mRNA, then via complementary base pairing, you're always going to get a specific mRNA sequence that corresponds to a specific amino acid, right? Because if complementary base pairing works and each amino acid is specific to a code, well, then you're always going to have the same thing move along, right? And so via complementary base pairing, you can create a specific link of polypeptides, of amino acids, which you can eventually be linked together to form a polypeptide. And all of this occurs on the ribosome, okay? So that's what we call translation. And it's based on the fact, on this genetic code, okay? Let's see here. Nice, fantastic. Okay, so one skill in the syllabus is to be able to interpret the genetic code. And I'm going to re-emphasize, it is specifically interpreting the code. So you do not have to be able to replicate the code, you just have to be able to know how to use it. And the way it works is basically that you just look at you find whatever code you're looking for, right? So for example, we could have CGA, and you know that CGA always corresponds to the amino acid arginine. And so when the mRNA CGA comes along on the ribosome, the tRNA that has complementary base pairing with CGA is going to be carrying the amino acid arginine. And so by linking these amino acids together, then what you can produce is a polypeptide, which follows a specific code and is kind of universal. Okay. Another application, or well, no, actually, these are just application, is that you have to be able to understand how Taq polymerase can be used to copy DNA a lot of different times. So Taq polymerase is just a specific type of DNA polymerase, which is resistant to heat. And so what you can do is you can basically speed up the replication process of DNA for a specific region of DNA to make a lot of DNA, and then you can compare this different DNA. Okay, but we're not going to go into more detail about it today because we sadly don't have time. Okay, and the other thing is that we can use bacteria to produce insulin. Now, the reason why we can do this is because the universal code, genetic code is universal. What does this mean? This means that all organisms, or most organisms that we find on the planet, use the same base sequences to represent the same amino acids. So that means if we can isolate the insulin gene in a human, right, and we can replicate this insulin gene, we can give this gene to bacteria and let them produce the protein insulin, right? That's quite neat. And the only reason this works is because the code for or the genetic code is universal. It works for all organisms, whether you're bacterium or you're a human. Okay. So yeah, these are just two applications you have to know. Fantastic. And are there any questions before we move on to cell respiration? No. Okay. Nice. Okay, so why is cell respiration important? Well, we need energy to stay alive. I don't have to tell you that. Or we at least need energy to not end up looking like this puppy over here, right? And the way that our body carries energy is in a molecule called ATP, or adenosine triphosphate, okay? And so basically, the main reason why we eat is because we want to produce these molecules of adenosine triphosphate. Okay. How does eating help with producing these molecules? Well, basically, the way we get ATP is by taking organic compounds, which in themselves have loads of energy, and then releasing their energy in a very controlled fashion so that we can produce ATP. And that sounds very abstract, but the main way that we do this is by cell respiration using carbohydrates. Okay. So there's two ways that we can do cell respiration. Oh, sorry. There's two ways we can do cell respiration. Yeah, exactly. Let me just delete that thing here. We can either do it in the anaerobic fashion or we can do it in the aerobic fashion. And let's first go through anaerobic. So anaerobic is the less efficient way to produce glucose, to produce ATP from glucose. But it does not require oxygen. Okay, so this is what your body does when it can't get a lot of oxygen, but it still needs to somehow produce energy. So this is what would happen if, for example, you're running a 100-meter sprint. Because as you're sprinting, your muscles are contracting so much that there's no blood supply to your muscles. And so the muscles don't get enough oxygen. So what do? Well, they know they still have to be able to run the sprint, right? And so they use the glucose available to them without oxygen. Now, this is okay in the short term because you only need to work without oxygen for just 11 or no, up to like 20 seconds or whatever, however long it takes to run a 100-meter sprint, right? But in the long term, it's not very efficient because you don't get a lot of ATP per molecule glucose, okay? So you have to know that, or you have to know two different organisms that can use anaerobic respiration. So yeast, right, the thing that you use to bake things, can perform anaerobic respiration, and in the process, it can produce CO2 and ethanol. And ethanol is the thing that you use to make alcoholic drinks. And in humans, glucose can be used to produce ATP by anaerobic respiration by producing lactate as a byproduct, right? So after you'll have done hard exercise, when you have that burning sensation in your legs or whatever, and that burning sensation is from lactate because it's actually an acid. Okay, so it's not very good for you. Glucose can also be used to produce ATP via something called aerobic respiration. The main difference here is that aerobic respiration requires oxygen, and it has a much higher yield of ATP per glucose. And so if an organism could choose, it would be much smarter to always do aerobic respiration compared to anaerobic respiration. Right? And anaerobic respiration produces CO2 and water as byproducts. Okay? Right, so you have to be able to apply this understanding to say that in yeast molecules, anaerobic respiration can be used to produce CO2, right, which can be useful when you're making bread and when you need dough to rise. And it can also be useful when you want to produce ethanol. So for example, for making alcohol, right, or alcoholic drinks like beer or wine. Okay, so that's done through what's called a fermentation process of glucose. And yeah, then anaerobic respiration produces lactate in humans. I don't have to go through that. You can measure how fast an organism is respiring using something called a respirometer. It's quite a simple device. It basically, what it does is that, or what happens is that you put an organism on one side on which you want to measure how fast it's respiring, and then you let it respire, right? So in the process, it's going to be using oxygen. And so as this oxygen gets removed from the test tube in which it's found, that is going to suck the air out of this tube over here, which is containing a liquid. Now, when you suck air out of a tube containing the liquid will want to move into that space where you've just removed the air. So if you imagine there being liquid along this tube, as you remove air, the liquid is going to move. And so based on how quickly the liquid moves, you'll know how quickly the organism is respiring. Okay? So I hope this makes sense to everyone. There's not too much activity on the chat today, but I guess I'll forgive that because we're not too many people. Okay. Right, awesome. So that's how a respirometer works. And you just have to understand that that's a way to measure how fast respiration occurs. The last thing is to talk about photosynthesis. Now, photosynthesis, right, is just the process, the reverse process of cell respiration. So in cell respiration, you use glucose and oxygen to produce CO2 and water, to produce energy, right? Photosynthesis is about using CO2 and water and energy from light to produce glucose and oxygen. So it's the way that the world ticks, right? It's a way to make sure that there's always enough glucose and oxygen available. Photosynthesis occurs in chloroplasts, and chloroplasts are basically contain these specific pigments in them which can absorb light energy to produce or to drive photosynthesis. Okay. Sorry about that. Okay, fantastic. Let me just close this. So, yeah, chloroplasts are the things that contain these molecules that can drive photosynthesis. And chloroplast is just another organelle, just like a mitochondrion is or a ribosome is. Okay? And the way that we have to the way that we have to the key understanding here in terms of the S cell component, because we sadly can't go through the HL, is to understand that photosynthesis is about making things from light. And there's two main stages to photosynthesis. There's what's called the light-dependent stages and the light-independent stages. The light-dependent stages occurs in part of the chloroplast, which we call the thylakoids. Okay? And the light-independent stages occurs in part of the chloroplast, which we call the stroma. Okay, now the stroma is basically just the cytoplasm of a chloroplast, right? But these reactions require adenosine triphosphate. Right. So photosynthesis, we can limit how fast photosynthesis occurs by limiting the amount of ingredients that we need for photosynthesis itself, right? So we said earlier that photosynthesis requires light and CO2, temperature, and also a little bit of water, right? And so the limiting factors for photosynthesis are generally described as light, CO2, and temperature and water. And what does it mean as a limiting factor? A limiting factor is something which prevents a reaction from occurring at a faster rate, right? So if I need CO2 to do a reaction, but there's not very much CO2 available, obviously I'm not going to be able to do that reaction very fast. So a limiting factor, yeah, is basically just something that prevents a reaction from occurring. We can't go through this, that's a little bit too much detail. Okay, the final learning point for photosynthesis is to talk about how the Earth's atmosphere has been influenced by photosynthesis. Okay, so the first time that photosynthetic plants came along was roughly, well, 3.7 million years ago, billion years ago, 2.7 billion years ago. And what you have to understand is that this is a really important moment in the kind of history of the planet, because all of a sudden you had plants that could produce oxygen. You had something that introduced oxygen into the air. But so what happens when these organisms first came to life on the planet? Well, what you might expect is that the oxygen simply began to build up in the oxygen. The oxygens began to build up in the atmosphere around it, right? But actually what happened is that the initial oxygen that was released from these photosynthetic organisms were used to react with the molten iron that was still present at the time, because Earth was still in its very early stages, okay? But so what happens is that as the photosynthetic organisms came to be, all of a sudden you had massive amounts of iron oxide starting to be produced, right? Because the oxygen was released into the atmosphere, and this oxygen was immediately reacted away with this molten iron, okay? And so at this point, you produce what's called iron band formations, which is basically just where huge deposits of iron oxide started to form at this level in kind of the Earth's core. So if you were to cut through the middle of the Earth, what you would find is these bands of iron oxide in different layers. And this was basically the start of the first photosynthetic organisms, okay? But then when did oxygen actually start building up in the atmosphere? Well, this occurred when all the iron in this very early stages of the Earth had reacted with the oxygen and had all been turned into iron oxide. And so basically all the oxygen couldn't react with any more iron. As a result, the oxygen could now expand into the actual atmosphere. And so this is when you actually had an increase in the levels of oxygen. Okay, so this is a graph to represent the oxygen levels on the Earth as time progressed. And although the first synthetic organism came to be around in the middle of stage 1, the actual increase in atmospheric oxygen was only seen a lot later, when all this oxygen has actually been carried out, or so when all the oxygen had reacted with the iron oxide, and hence there was no more iron to react with oxygen. Okay, exactly. And so that was just one learning point. And then the final learning point is just to understand that the pigments that we find in plants to do photosynthetic, the pigments that we find in plants found in these chloroplasts can actually be separated out, and we can analyze their different structures. And we do this via something called chromatography. And chromatography is just a technique to separate different substances. So what you do is you basically just dissolve whatever substances you're interested in the liquid down here, and you place what's called a chromatography strip into this liquid. Now, because water which is the thing that is dissolving all the different pigments, is very good at dissolving things, right? We talked about that earlier in subtopic 2.2. The water is going to carry all the solvents up this chromatography paper, because water obviously also has adhesive properties to this chromatography paper. Okay, so you dissolve things in the liquid. Water has adhesive properties, which means that it begins to travel up the chromatography strip. And in the process, it carries along with it all the different molecules that make up the chloroplast. But as the water continues to travel up this chromatography strip, it's going to get, or the heaviest pigments that are dissolved in this liquid, in this water, aren't going to be able to travel very far. So around the beginning of its travel, water is going to get rid of its heaviest molecules that it's dissolved. And so, as you can probably imagine, as water continues to travel, you'll have separations in terms of the different liquids, or sorry, in the different pigments that are contained in the chloroplast. So when you're done with chromatography, it's basically a way to separate out different pigments. Now, you might have done this experiment in class. I'm not sure if you did, but you just have to be able to understand how it works. And so hopefully that should work. Okay. Fantastic. And so that is basically the end of topic 2, molecular biology. Does anyone have any questions about any point in topic 2? We have someone joining us. Okay, well, that's great. So no questions is, I guess, always a good thing. Well, then, if no one has any questions, then I would like to wish you all very good luck on your exams, and hopefully I will see you all very soon. So yeah, that was basically it. Just do send an email or anything like that if you have any questions at any later point. And otherwise, yeah, best of luck with everything. Bye-bye, guys.